All right, so this is the final biology of cancer. How is cancer treated? Well, mutations in cancer-causing genes can be tested for. So what we can do is we can actually get down and look at your DNA. It's isolated from white blood cells. We put it through this thing called polymerase chain reaction. And what PCR does is it makes copies of the gene of interest. What we'll do is we will, heat the sample to denature DNA. And what it means is instead of it being double-stranded, we'll pull the strands apart. And then we'll cool it down to lower temperatures, so from 95 to 55, to put in these things called primers. And what these primers do is they're looking for very specific sequences of DNA that we're looking for maybe in these cancer-causing genes, and then that will get copied, and then at the end of cycle 1 you have 2 copies, end of cycle 2 you have 4 copies, end of cycle 3 you have 8 copies, and we'll do this for about 40 or so cycles maybe, or even more. It just depends on what you want to do. But if it doubles every time, then you could end up with millions of different copies of DNA. And then once copies have been made, the exact sequence of nucleotide is determined through sequencing, and so we can add tags to the nucleotides, run them through a laser, and it identifies the ones that have the fluorescent tag, and then we can see the actual sequence of G's, T's, C's, and A's, and we can compare that with known sequences to determine if any mutations are present or not. And if they are, then we can begin to look for the evidence of cancerous cells. Gene chips, or these things called microarrays, can screen genetic information too, allowing doctors to look for specific mutations. Synthetic DNA that corresponds to known cancer mutations is placed on a chip, like a computer chip. Then the RNA is isolated to create labeled DNA. And once it hybridizes with the synthetic DNA, then the chip can alert the doctor that mutations are present. Blood tests called CBCs look at the presence of normal red blood cells and basic blood chemistry. And the reason is that cancer can elevate the number of white blood cells, and tumor cells may even be present in the bloodstream. If you have changes in calcium, enzymes, or certain ions like potassium or sodium, it can also indicate that cancer is interrupting the function of an organ somewhere. And so it's kind of like a clue that there could be cancer present. If these things are present, it doesn't necessarily mean that it is cancer, but it's just potentially caused by cancerous. We can do mammograms, CT scans, PET scans, MRI. You can check out those things. Make sure you do your little quick check. Well, a lot of times when we hear the word cancer, we hear, chemotherapy associated with it. Well, what is chemotherapy? It's the use of drugs to treat disease. And in the case of cancer, chemotherapy works by targeting this cell to prevent cell division. One of the drawbacks of chemotherapy is that it affects all the cells of the body. So it can be effective in treating and preventing metastasis, but it also has a lot of negative side effects because it keeps the rest of your body's cells from dividing too. And so you have this kind of whole system effect, but, chemotherapy hopes, the doctor who supplies the chemotherapy is hoping that they can kill the cancerous cells before the entire patient has too many side effects. So you have alkylating agents. These are chemicals that integrate into the DNA, causing it to break at specific points. It can cause long-term side effects, including other cancers. Alkaloids are produced by plants, and they prevent cells from forming those spindle fibers, preventing cell division. But again, that affects your whole body too. Taxanes prevent microtubule breakdown during cell division, so the cell division doesn't continue. Anti-metabolites mimic nucleotides and are stuck into the DNA, but it interferes with replication, therefore preventing cell division. And then topoisomerase disable this enzyme that prevents DNA replication too. So all of these things are designed to keep rapidly dividing cells like cancer from dividing so much. So how's radiation used to treat cancer? Well, radiation is the movement of energy in waves or particles. So light is radiation, radio waves are radiation. And the shorter the wavelength, the more energy that's present. And radiation uses high energy gamma rays and X-rays, including alpha and beta particles, which are things found in the atom. And what we want to do is you want to kind of hone in on the area where you find a tumor. You don't want to radiate the entire body. So chemotherapy is a whole body therapeutic, but radiation is designed to target specific areas. So it's not really good against metastasized cancer cells. Radiation therapy is good for if you find a place where a tumor is and it's the only tumor that's there. Now, radiation is dangerous because it can cause mutations in the areas around the tumor too. So doctors have to be pretty precise with radiation therapy as well. And what are some future of cancer treatments? Well, the immune system is one of the things, because the immune system is kind of designed too to help to identify cells that are dividing uncontrollably and to kill them off. And so therefore, some cancer vaccines use modified viruses to initiate immune responses against cancer cells. You can create antibodies to target specific cancer antigens. This kind of is a visual to this slide here. We can edit the genome in certain ways to look for specific sequences. And then there's this thing called CRISPR, which is a brand new, well, not brand new, but it's a fairly new discovery. It's clustered regularly interspaced short palindromic repeats. And it was found in a bacteria. And what CRISPR can do is it can remove nucleotides, inactivating the gene, or it can insert new ones, which changes the function of the gene. So we can take out those gene controls on those oncogenes and replace them with something else, and therefore turning off those oncogenes. This would be CRISPR-Cas9 kind of a diagram. So this is the complementary to the target gene. This is what we want to have. This is a guide DNA. You put this in the Cas9 enzyme. Here's the target gene here. The complementary on the guide comes and finds the section of DNA we want. It will cut that out, and we can either delete it or replace it with what we put into this CRISPR-Cas9. And that is another kind of short chapter for how cancer is treated.